This session is a continuation from our therapeutic development session from yesterday. Today's focus is going to be on large molecules. So we will hear about gene therapy, mRNA-based therapy, as well as enzyme engineering. And to kick off this session, I welcome Dr. Gerry Lipshutz to the stage. Dr. Lipshutz is a physician scientist at UCLA. His work focuses on developing gene therapy for rare metabolic and neurodevelopmental disorders. He has done extensive work in the areas of arginine metabolism and more recently in creatine metabolism. His work bridges basic science as well as clinical applications. Dr. Lipshutz, all yours.
Thank you. This is the, okay, thank you. Okay. Good morning. So there's a lot here. I prepared this talk for the parents, so for the scientists in the room and the clinicians, so please excuse me if it's missing some details. So having been here these days, and you all having been here these days, some of the topics that I present briefly, you've heard already, but maybe it will further emphasize them because my slides were made independently of the people who presented previously. And I think Dr. Kakis spoke a little bit about this. I like this idea of this concept that creatine is considered a battery for regenerating the source of energy in cells, which is ATP. So ATP, adenosine triphosphate, is the source of energy that all our cells use. And certain types of cells or organs require bursts of activities, such as the brain, the heart, skeletal muscle. And that's why we see these deficits in those organs with creatine disorders, because there's not enough energy. So if you can think of it as creatine phosphate is really the battery that regenerates ATP. It takes it from ADP, which has two phosphates, and adds a third, taking the phosphate from creatine phosphate, and then creatine phosphate adds that phosphorus back to ADP to make ATP to make energy. So if you don't have the battery backup, you can't make enough energy. And that's why we have these disorders, if you think of it that way. So I'm going to talk to you about the work that we've been doing, an update for some on the work we've been doing in creatine transporter disorder, and then follow with some update on GAMT. And this is in our work in gene therapy, which is the area that I focus on. So this was touched upon, but I really want to make sure that it's clear to everyone about this concept of the blood-brain barrier. So the blood-brain barrier protects the brain from toxins and infectious agents by tightly regulating the passage of chemicals between the circulation and the brain. Not only does the blood-brain barrier prevent most molecules from interacting with the brain tissue, but the blood-brain barrier prevents drugs, it prevents chemotherapies, immunotherapies, antibodies, gene and cell therapies from entering the brain. It really is a mechanism of preventing the brain from being injured by substances we might be encountered to in our environment that might enter our body. So it's something that's protecting us. But in order to deliver something to the brain, we have to somehow cross that barrier. Or at least by what we're doing, we need to learn how to cross that barrier. And creatine enters through the transporter that's in that barrier. It exists there. So creatine, a valuable substance, can go across that barrier naturally. And then the transporter is also within the cells in the brain, the glia, the oligodendrocytes, the neurons, to bring the creatine into the neurons. So you need the transporter both in the barrier and in the cells in the brain. So these are really the challenges or the questions. How can we achieve and normalize brain creatine levels? And second, how can a method for restoring functional creatine transporter in brain cells be accomplished? So people have tried to deliver gene therapy agents to the brain by different mechanisms. So what's a gene therapy agent? So what I'm talking about today, and what probably about 90% of all gene therapy trials are using a virus that exists, that we've taken the genes out of that are part of the virus and put therapeutic genes in. So we essentially eliminate the viral genes and put in the therapeutic genes, and then deliver them by some mechanism to the brain. And what people have done is, if you have a certain area of the brain that maybe that you want to deliver it to, you can make a hole in the skull and inject a needle into that location under some radiologic guidance, but it's quite limited to the area that you're injecting it to. Generally smaller than this area that's designated in this picture. Others have tried to deliver it to areas in the fluid that surrounds the brain or the spinal cord, in the lower back, in the lumbar intrathecal injection, into area in the back of the neck called the intracisterna magna injection into the fluid that bathes the brain, or through the brain into an area of the ventricle in the brain to deliver the viral vector or a different gene therapy agent to the brain. But there are some limitations with these because they are invasive and they generally have more limited spread of the gene therapy to the entire brain. And as we know, these disorders, particularly creatine transporter disorder that I'm talking about in this first part of this talk, affects the entire brain. So we really need to get all the cells to express the new gene. And these mechanisms tend to be limited in what they can express in the brain. So we're using this concept of delivering intravenously, like how you might have your blood drawn in a vein in your arm, but administering it this way, the gene therapy, by some advances that have gone on recently in the development of these viral vectors. So this is why it's important to understand this concept from the beginning for creatine transporter disorder. So these are some pictures here of mouse brains, okay? This is a mouse brain where a green protein called green fluorescent protein is expressed in neurons, in the nerve cells. And that's what we're seeing here in this blow up. In here, we see blood vessels throughout the brain. And you see this density of blood vessels. When they are overlapped here, we see them together. The blood vessels and the nerves, the neurons, are very intimately related. So that most brain cells are within 20 to 30 microns of the nearest capillary. So if we can get a gene therapy agent into the blood vessels that are near the neurons and it can cross, you presumably could have that vector or have that gene therapy agent express in all of the cells of the brain, which really is, I think, the goal for CTD. So here's a diagram of a gene therapy vector. We use, which is commonly used today, is something called an adeno-associated virus. If you have a dog and you vaccinate your dog for something called parvovirus, this is in the same family as parvovirus. It's not the one for dogs, but it's in the same family. You've probably heard of a parvovirus before. So here's the genome, the genetic material, and here's a blow-up of the genetic material. We remove the viral genes, and you insert your gene of interest, in this case the creatine transporter, with regulatory components, and the only parts of the virus that you keep in the genetic material are these ends called ITRs, or inverted terminal repeats, that help package the genetic material within the protein around it that allows it to infect cells. I hope this makes sense so far. So when you deliver it to the bloodstream, the goal here is to use this mechanism here. It's in the blood vessels. Here's the blood vessels here. Here's red blood cells in the blood vessels, and here's the viral particles. But we need them to escape the blood-brain barrier. And we're using this technology, this advancement, where something called transcytosis, which has been used for certain drug delivery to the brain, where the viral vector can bind to the membrane of the blood-brain barrier, go through the cells, come out the other side into the brain, and then those viral vectors, because they're so close to all the brain cells throughout the brain, can infect those cells. The word is transduce, but you can think of it as infect those cells and deliver the genetic material to all the cells of the brain, or essentially all the cells. That's the concept that we're trying to generate with CTD. So we've generated these types of viruses and tested them in mice. And here, in these pictures, we're expressing that green protein. The images don't look green because of the way the study is done, but it's a green protein. It's not the creatine transporter. And here we have 7 different types of viruses that we generated. Again, the adeno-associated virus have different protein on the outside. Some work better, some work not as well. So this is what it looks like. There's a mouse here, but you don't see it because the virus didn't get into the brain. In this case, we're just using salt water, what we call the vehicle. But all these 7 other ones are different viruses injected into the mice, and then these mice are imaged a certain way so that we just see the brain and spinal cord. And that's what you're seeing here. So in these 4, this one here called C, D, F, and G, we see the brain and we see the spinal cord with expression of this surrogate protein, this green fluorescent protein that we would substitute out and instead use the creatine transporter. I hope that makes sense. It's easier for us to see and measure the activity of the green protein than it is for us to do with the creatine transporter. So the one that we've been really focusing on is one we call F right here. So then we've taken these mice, they've been administered these viral vectors when they're 30 days old, we euthanize them 4 to 6 weeks later, and we actually take out the brain and we section the brain, and now we look for that protein, the green fluorescent protein that glows under specific light. Here's the one with just salt water injected. You don't see anything, but you do see in these brain sections, how the brain is cut from the mouse here, you see this expression in at least 4 of these. And again, this one I put in red is called AAVF. We call it F. And that's the one we've been focusing on. You can see here we can quantitatively measure the amount of this green fluorescence. Some have 0, some have a lot, including this one called F. So Alex Boganowski from my lab mentioned this yesterday to people, and I know not everybody here is a molecular biologist. So there's this concept in biology that you have DNA in your nucleus of your cells. That DNA is then made into something called RNA, which is the message. And that message is in the cytoplasm outside of the nucleus, and it is the message or the instructions for the protein to be made. In this case, the creatine transporter, and the gene being SLC6A8. So the light is a little bright, but what we have here is a section of the mouse brain, and we made a probe, a piece of genetic material with a red fluorescent protein stuck to it. And when we have euthanized this mouse, took out the brain, put sections of it on a slide, added the probe with the red dye on it, it's made to match this genetic material. It's designed that way. So we can see that throughout the brain of this mouse, throughout this section of the brain of this mouse, that probe has bound to the genetic material that's been produced by the virus. It's not the virus DNA. The DNA now has been incorporated into the machinery of the cell, and it's producing the instruction. And we see this red probe throughout the brain, telling us the instruction is in these cells to make the transporter. So we found, and along with the Sara's group has found this too, that we can use a type of imaging of mice that people have called a PET-CT. And what the molecule we use here is related to a sugar molecule, glucose, and it helps us determine the metabolic activity, how well the brain is working or how well the brain is using sugar. Now, as I mentioned at the beginning, we have 2 sources of energy for the brain. We have creatine as creatine phosphate, and we have sugar or glucose. This is looking at glucose activity. If you don't have enough creatine or creatine phosphate, you're going to preferentially use glucose because you need to make energy. So what this shows us here, if we look at the brain in the oval here on a normal mouse, a wild-type mouse, you see mostly this green color here, green and some blue, demonstrating a certain level of activity of glucose metabolism or sugar metabolism. If we look at the untreated creatine transporter mouse, we see there's more activity of the red. It's actually using more glucose, more sugar than the wild-type mouse because it doesn't have creatine phosphate. If we look at our treated mouse, again, just in the oval here, it looks much more similar to the wild-type mouse, showing us that we've been able to alter the metabolic activity, and we see this quantitatively here. So the gene therapy has suggested that we have a change in the metabolic activity closer to normal. But what's important is behavioral activity. Can we change the behavior of the mice? So mice have normal activities that they do. If you add something novel in a cage, like a marble, it wants to bury it. That's what mice do. They bury it. Or they make a nest. If you add certain cotton or newspaper, it'll tear it up. It'll make a nest. It turns out that CTD mice don't do those activities, or they don't do them very well, or don't do them at the same rate as wild-type mice. So if we look over here on marble burying, here's a wild-type mouse, or a group of wild-type mice. This is how well they bury marbles. The mutant CTD mouse doesn't do that. It doesn't have the activity here. When we do perform this gene therapy, we see some improvement in that activity. I mentioned that they like nesting activities. So we add it's like a firm piece of cotton called a nestlet. And again, normally mice will tear this up within a couple of hours. Here's a 5-hour interval picture. It tears it up to make a nest. The CTD mice generally don't do that, or don't do it as quickly. With the gene therapy, they in fact return to have that activity. It's restored. So we can see here also, it's not the same as the wild-type mice, but it's certainly improved over the mutant mice. This was talked about a couple times already. Just as a reminder, looking at the creatine peaks in MRS, here's an article published by Clark. These are taken from there. CTD male, this creatine peak is very small. In a GAMT female, the peak is very small. With creatine for the GAMT patient, you see the peak restored. So we employed this technique to our mice with our colleague at University of Minnesota, Pierre Gillies-Henry. His name has been mentioned a couple times here. And I just want you to look at this here. So this is not our optimized vector. You start with these projects, you start with a multiple of different ideas. You generate multiple different viruses, and you try to funnel down to one or two in the end that you're really going to focus on, that have the properties that you want them to have. This was not one of our best vectors. But I want to show you the potential here. So here's an untreated mouse, okay? A creatine transporter, untreated mouse. This is its spectrum that now we've seen multiple times. Here, this is the minimal creatine peak it has. It really has nothing. Turns out there's two peaks. There's one over here, and there's one over here. This is what you would have in a CTD mouse. This is what we have in a CTD mouse. This is a normal mouse, a wild-type mouse. So look at this peak here. Look how big it is compared to this peak in the mutated mice. Look at this peak here again, its absence, and here it is in a wild-type mouse. In our treated mouse, again, not using our optimized vector, here's a peak, partially restored. Again, compare them to the others. Here's the peak over here, partially restored compared to the others. So not using our optimized vector, we're able to go 53% of wild type with this method of analysis. If we then look more quantitatively, so this is the data quantitatively. I'm not just pulling it out of the air. It was analyzed. It's about 53% of normal. As I showed you on the picture a short time ago, looking at the red dots, looking at the message, now to look at the actual protein, the actual transporter by a method with an antibody. Here is, you can't see because of the lighting here, but there's mouse brain section on this slide. Then we used an antibody against the transporter, against the protein itself, and here it is here. So we now know that we have the transporter in all of these cells throughout the brain. If we look over here, we took out the brains and we measured the amount of creatine. This is from a very high dose called 1 × 10⁻⁴, but we see a dose response. Here's the mutated mouse without treatment. Here's a wild type group of mice. And we see that as we give more of the viral vector, in general, we get more of the protein present and more creatine present in the brain of the mice. So for the last few minutes, I want to talk a little bit about what we're doing with GAMT and what we've progressed with here with GAMT. The approach is different. As you've heard in the last couple of days, the GAMT enzyme is mostly in the liver. So our therapy has been directed to the liver. So we've modified the virus that we use, more of a traditional adeno-associated viral vector, and we don't have the SLC6A8 gene, we have the GAMT gene. We have the regulatory elements that turn on the expression of the gene. We have it in a virus. These viruses are made in billions and trillions and quadrillions of copies of these viruses, and we direct it intravenously so that it goes to the liver. And here we gave a dose of 5 × 10⁻³ viral particles per kilogram. A mouse doesn't weigh a kilo, it weighs about 25 grams, but that's how we do the dosing. And these are 3 separate mice, 3 individual mice that have been euthanized. Their liver has been taken out, and we've looked for the GAMT protein. And we see a little bit of differences between them. Here, all the brown pigment you're seeing is expression of GAMT. There's hundreds of cells here that have GAMT. These more white cells right here really have probably little GAMT expression, versus the really dark cells have really high amounts of GAMT. Here's another animal. There's a lot of very high expressing cells, and there's some cells that are expressing very low or little. And then here we have a little bit more of cells that have all different expression. You have some higher cells, you have some more that are moderate, some that are lower, but we get a lot of expression throughout the liver. And because we get a lot of expression throughout the liver, we again see changes in the brain because we're restoring creatine in the brain, which I'll show you on the next slide. But we see that metabolic activity again is altered. Here's the wild type. We have more red and yellow. Here's the untreated. We have very little of that, but then it's restored to more like the wild type with the gene therapy. And that's expressed quantitatively here also. You can see it with this PET imaging, this glucose activity imaging. And then here again, we've done the MRS with Pierre Gillies at University of Minnesota. And again, if we look at the wild type mouse, here's the creatine peak, one of them. Here's the other one, creatine peak here. If we look at the GAMT deficient mice, and these GAMT mice, we use vegetarian food for them, vegetarian chow. It actually exists for mice. So that, because regular mouse chow has actually a little bit of protein, it has fish meal in it, and it causes their creatine to go up. So they have no creatine being given to them. And this is what you see in these mice. They have no supplementation. You see this tiny little peak, if any, and tiny little peak here. And this is what we get with the gene therapy directed at the liver. We get this peak here, again, compared to the 2 above it, and we get this doublet here, peak here, again, compared to the 2 above it. And calculating it out, Pierre Gillies determined this in his team, it's about 80% of wild-type creatine levels in the brain. With one, this is one injection. This is not multiple injections. This is one administration only. And it hopefully will last a lifetime, if not a very long time. This is it quantitatively. So we don't, with people, you're not doing brain biopsies to determine how much creatine is in the brain using the MRS. We euthanize, but in people, you take plasma, you take blood, and you measure it in the blood. So if we look at the creatine levels, here's the mutant creatine, very little creatine in the plasma of these mice. This is wild type, normal levels. We see as we increase the dose of our viral vector, we get up to normal amounts of creatine in the bloodstream. We can normalize it. There's a dose response. Same for GAA, a toxin. It's very high in the mutant mice. Here's in wild-type mice, less than 5, which Dr. Longo spoke about as a goal. And this is what we see. We see a decline in GAA to normal with the higher doses of the viral vector. The imaging I showed you on MRS, this is the quantitation of the data that Pierre Gillies put together. That's about 80% of wild type. But then when we actually take out the brains of different doses, we also can actually measure the amount of creatine, physically measure it. And again, we see this dose response. You give more, you get more creatine in the brain. So this is the final part of my talk. This is the pathway to take it forward. In this country, we go through the FDA, the U.S. Food and Drug Administration. And again, it was touched upon a little bit, I think, by Emil Kakis yesterday. But you start with a proof of concept. You've demonstrated something in mice or in a different model that works. So then we started with a funnel of different viral vectors. We narrowed this down. We identified the final therapeutic candidate. We've done that with GAMT. We have the funding to go quite a bit of ways into this that I'll talk about in a moment. But you identify your final therapeutic candidate. You then have a meeting with the FDA called an INTERACT meeting. This is often a written meeting. It's not necessarily a Zoom meeting. It can be a written meeting. And I'll tell you about what we've done already with that. And then you have that meeting. Hopefully everything goes well. Then you have a scale-up of the production called CMC that, again, Emil Kakis spoke about yesterday. And this tends to cost a lot. And then you do these studies in mice. You make what's called a GLP vector, a good laboratory practice of your product that you want to give to people. It's almost at the scale you could give to people. It's a little bit less than that. But you test that in the animal model for efficacy, but really also for safety, because that's what the FDA really focuses on, is safety. They don't want anything going out there that's going to hurt anybody. That's the most important thing, is safety. So you do these studies in mice, looking for safety. You prepare all this as a document. You submit it to the FDA in what's called a pre-IND meeting, pre-investigational new drug meeting. And then you hope everything goes well with that. And then they might request, or you might want to do some additional testing with your viral vector in animals. That's what I have here. You then also, at that point, you make the vector for the clinical trial. You generate the vector for a clinical trial, and then you develop your clinical protocol, and then you submit an investigational new drug application to the FDA, and they have 30 days to respond. And if they don't respond in 30 days, that's basically giving you approval to do your clinical trial. And then you do your clinical trial, and some of these can be combined. Phase 1, phase 2 could be combined, and then phase 3. Sometimes you even have a phase 1, phase 2, phase 3. But phase 1 is the safety and dose determination. You already have some idea of the dose because you've done these studies in animals. It's not like you're going in blind on what the dose should be. You know from other trials, you know from what you've done in animals. So you have phase 1 and phase 2 is efficacy, and then phase 3 might be more of a confirmation of the dosing of the dose you want to give, and then you have probably some process performance qualification on the product that you have to go through, and then you file a new drug application and you move forward into treating everyone with disorder. So this is sort of how it works. I don't want to go long here, but this is what we've had with FDA interactions already. So for GAMT, we obtained orphan drug designation through a process with the FDA. We also obtained rare disease designation, which are important things that will help us bring this forward to patients. We had our INTERACT meeting, and we hope to have a pre-IND meeting in the second quarter of next year when we get through a lot of these studies that we have remaining in mice. For CTD, we've obtained orphan drug designation. We've had our INTERACT meeting request, and we also obtained something that the Trump administration put forward. This is not a political statement. The Trump administration has actually tried to help the rare disease community with this program called Rare Disease Evidence Principles. We applied to that program, and we actually got accepted to that program for CTD. We are in the process of submitting this rare disease designation application like we have for GAMT. It'll be submitted hopefully in the next month, and then we hope to have a pre-IND meeting for CTD with the FDA hopefully next year. So why the RDEP program is so important is it really fits CTD. When you read this, you say, this is CTD. A target population less than 1,000, a genetic defect that's an inborn genetic defect that drives the disease pathophysiology, that there's disease severity, it causes progressive, rapid, and significant disability in a short period of time, and there's no alternatives. This really fits the program. The FDA recognized that and gave us the RDEP designation to proceed with, which allows us to have much more communication with the FDA to hopefully have one appropriately controlled clinical trial for this type of therapy to move forward if everything continues to go well in mice. So I just want to thank all the people who have helped us, particularly Andreas Scholze and Alanna Takikova at Sick Kids in Toronto. We couldn't have done these studies without them. From the University of Minnesota, Pierre Gillies and his team, Pierre Gillies Henry and his team who's doing the MRS for us. The funding that we have from California Institute of Regenerative Medicine. We've had these partners who've helped us from both the imaging studies to our regulatory support, and then our funding from California Institute of Regenerative Medicine and SK Pharmteco, who's been our biotech partner generating our viral vector for us. Thank you. I'm open to any questions in the time that might remain. And I know that was a lot to take in. I really just wanted to give you a flavor of it, and I think a lot of these things were spoken about already.
Thank you for this amazing talk. What duration, how many days were there between two spectroscopy exploration?
The mice were injected, and 3 months later the spectroscopy was performed.
Okay,
we only did it once on the mice.
Okay, and another short question. On one of your slides, you showed that the amount of creatine was even very high. I mean, higher than expected,
Yes.
Higher than the wild type.
Yes.
Do you have any concern regarding that?
Yes, that was on one of the earlier vectors. On the new viral vectors we've developed, we have levels that are closer to wild type that are not exceeding, because I'm not sure that exceeding wild type is a good thing. Exceeding normal levels of creatine in the brain may not be a good thing.
Thank you. That was interesting. Do you see the same behavioral patterns with CTD, with the marble burying and the nestlets for GAMT or for AGAT mice?
So we don't study AGAT. For the GAMT mouse model that was developed in Germany, Dirk Isbrand, in the original publication, they didn't really find too many deficits in those mice behaviorally. In our mouse phenotyping core at UCLA, the scientists there, through that studying that article, helped us identify some additional deficits that the mice have that we use. I didn't present any of that here. We've published on that previously on the deficits and the resolution of those behavioral deficits in the GAMT mice with our gene therapy approach.
Thank you. Also, and I noticed you use the word restore a lot in terms of brain creatine peak, and I'm assuming that's to wild type levels, whatever the variation is. But I'm wondering if you've correlated any of the creatine peak restoration in terms of the area under the curve with other biomarkers, for instance, plasma or urinary creatine levels?
The area under the curve for the MRS? Yeah. We have, I could only put so much here, and I don't want to overwhelm everyone, but when we study the urine for the GAMT mice, the creatine levels are normalized and the GAA levels are essentially non-existent. Well, you know, these studies are really expensive, particularly the MRS, and we can only, you know, we have problems shipping mice to Minnesota because of weather at times. So I can't say that we've done that. We have not. I don't know why they would not correspond, but we've not done that specifically.
Thank you for all your research and the status. I have two questions. So first is that the RDEP designation, I know you mentioned that for CTD it was recently kind of classified or approved. Is the GAMT already in that RDEP category or
No,
not?
they declined for GAMT. We applied for GAMT also, but they did not issue it. We're going to appeal, but I don't know that it'll be reversed. But we applied for that too.
And did they give a reason that why it does not qualify? Or
the reason I think was that there's alternative treatments that have some level of effectiveness. You know, it's hard to fight these battles. You know, there's again, it's not a political statement. It really is not meant to be. There's been a lot of changeover in the FDA. You know, our regulatory member Tim Cote from Only Orphans Coat has helped us. He worked at the FDA for 20 years, and he's helping us with this. And he says we should appeal, and he'll help us appeal. But I don't know that it'll get reversed.
Got it. And just follow-up question that in this status for Q2 2027 for Interact meeting, from your previous chart, how much time does it generally take from getting Interact meeting to phase 1 trials? And rough timeline, I'm just talking about.
So the question is, how much time from an Interact meeting to get to a phase 1 trial, assuming the studies continue to go forward in a positive direction, right? Yeah, you know, I'm sorry the way this sounds. In the end, it really comes down to money. For GAMT, the process is clear. We're going to have a pre-IND meeting. We have all the money to get us there. Again, I'm not saying this because I'm asking for money. I'm just telling you how it is. We have all the funds. We're going to have a pre-IND meeting. There's no question about it. Because we have this grant from California Institute of Regenerative Medicine. But for CTD, we don't. That's ending at the end of October and is a much smaller award. So if everything works right, you know, when could be in a trial for GAMT for this? It's hard to say exactly, but probably within 2 years if we had the money. Again, it's not an advertisement for money. For CTD, there's still some steps that need to happen to prove safety, to ensure safety before it can go ahead. We've not seen safety signals in mice, but the technology that we're using for GAMT has been applied multiple times to human clinical trials. For GAMT, it has not. For CTD, it has not. And another group with another genetic disorder will likely be using the same technology for a genetic brain disorder in humans in the fall, probably in the fall. And if that turns out to show safety, then I think it opens a big window here, if there's funding.
Yeah, thank you. Interesting talk. I'm over here, right next door.
Oh, okay.
While you didn't elaborate on what actual capsid addition you made to mediate the RMT, is it more some of the ones that have been used in previous attempts to get across the blood-brain barrier?
It hasn't been.
Okay.
It hasn't been used clinically yet.
And as given it intravenously, do you see activation outside of the brain?
So our method for restricting is primarily based on the genetic regulatory elements for the families, the promoter portion.
Right.
And that's how we're restricting the expression. You know, these have been, what people don't know is that this has been very challenging experiments. The CTD mice are very thin and frail. And you say to yourself, should I try to develop an approach that's going to correct the abnormalities in the heart that we found, the abnormalities that some people here have talked about in their children, and the decreased muscle mass, or should I just focus on the brain? And it's difficult because the mouse is different than the people. And this was talked about by Laura Baroncelli a little bit the other day. And it generates questions in you because sometimes you say to yourself, when you do the therapies on the mice, are the mice not, the treated mice, are they not able to accomplish the task because there's still a problem in the brain or because they're weak? And they are weak. They often can't find the food. If you have the food in a different location, a different cage, it's hard for them to find it in the untreated mice. It's just, they can't reach it. So there's challenges with the model. There just is. And so we are trying primarily to focus on the brain for the treatment with a little bit of expression in muscle, which would be hopefully help with, I think, what is an issue in the intestine, the constipation and GI issues that the parents report all the time, that hopefully maybe it will help with that. It'll hopefully help a little bit with the heart and with the skeletal muscle, provide energy also.
So then as a follow-up and maybe as a lesson for other people that are considering gene therapy for the transporter being a transmembrane protein, at the Interact meeting, was there any discussion about immunogenicity risks?
There weren't. The thing with I just tell you straight out, with the Interact meeting with the FDA, it depends how you want to interpret the response. The response was, because it's all written, says, you're far enough along that you don't need an Interact meeting, you need a pre-IND meeting. You need to be at the next step. They already thought that we had done enough, that we should have the next step. At least that's how the positive way you could interpret it. Then they had a bunch of questions that they said we should address in a pre-IND meeting.
Was immunogenicity one of them?
No, it wasn't. It was not.
Is it for you, though?
Immunogenicity for the transporter?
You're essentially introducing a new protein, a foreign protein in the body. And as much as you're protected by the same blood-brain barrier you're trying to get across for the brain, if you have expression in other tissue sites, you could be promoting an anti-transporter response.
Well, yeah, this is something that probably needs to be thought a little more about. We'll see what happens with the groups that are ahead of us that are going to use this approach. But for example, SMA, spinal muscular atrophy, 4,000 children have been treated with AAV9 expressing SMN. And some of that, they get administered vector early enough that some of it's entering the brain. And so far, there hasn't been a concern that way. Now, it's at much lower level of expression than what we would have with the creatine transporter. So far, we've not seen safety signals in mice that suggest problems. But these are things this is why I said we need more time to work out for SLC6A8. It needs more time. It needs more studies. We need to make sure that it's safe.
So we have a couple more questions, but what we also need to do is a computer swap for the next talk. So maybe if I could have you just step down from the podium with your mic set, and we'll do the computer swap and take those additional questions.
I'll answer them from over here.
Hi. Yeah, thank you. Question about the AAV for the CTD that's crossing the BBB. Do you anticipate needing to do NHP studies? And in light of the capsida safety event, where I think they didn't see any signal in their NHPs but did see safety in humans, do you think there might be some concern about the target of the capsid and maybe differences, species differences in binding?
So the approach that we're using, we're working with another group, it turns out it won't, so NHP stands for non-human primates, it means monkeys for everyone here. This approach, and this is some of the beauty of the approach, is that it won't work in monkeys. It will only work in mice that have been designed, that have this human receptor in it, a knock-in mouse, or people. So it eliminates the testing in monkeys. The group that designed this has already gone to the FDA and has approval for what they're doing to move from mice to humans without another animal model in between. So it's a different approach. I do understand your question. I do understand the concern, but there isn't another model to test it in. So we have to be extensive in our testing in mice. But again, and I don't mean it the way it sounds, we're not if this proceeds and we have the funds to have it proceed, we're not going to be the first people that are testing this approach in people. It's not going to happen. There's going to be another group that's probably going to test in the fall, and there's a company that's been developed for some other disorders that will probably be there before us. So that, again, I don't like the way it sounds, but the safety of this, in some ways, some of the safety of it will already be tested for other genetic disorders before we reach that stage.
Along the heels of that, maybe one more question. You mentioned that there's another disorder, brain disorder, that is currently testing this vector. Are you able to shed light on which disorder this is, so we could perhaps have learnings from those types of studies?
I can't, but I know there's at least one person in this room who has been involved, been connected with them, that could if they want to, but I can't. It's just how the rules are, the agreement that we have. You know, what you all don't realize is that not everybody's altruistic on these things. You know, the companies protect things, other family groups protect things, and we're held by these non-disclosure agreements. And I don't want to jeopardize any program that we might be able to have for these patients by not sharing something that you might want to hear, but I just can't at this stage. I don't really know anything. I just know the group's name. That's it. There's really not hiding anything from you, but that's the bottom line.
Thank you so much, Dr.
Thank you.
Lipshutz. That was an excellent talk. Thank you for your time. It's my pleasure to welcome Alanna Schepartz to our meeting. This is her first time at this meeting. Dr. Schepartz is the CZ and Irmgard Chu Distinguished Professor in the Departments of Chemistry and Molecular and Cell Biology at UC Berkeley. Her research group studies the chemistry and biology of complex cellular machines and exploits this knowledge to design molecules. She has won numerous awards. She is also a member of the National Academy of Sciences. And another addition to her cap is that she is also one of our Race for a Cure awardees. We specifically looked at the technology that Dr. Schepartz was developing because we thought it would be applicable for GAMT. She very kindly indulged us in this thought exercise and then submitted a proposal to develop the same. So today she's going to share with us some of her recent discoveries and specifically talk to us about the technology that she's developed that may enable these efforts. Thank you, Dr. Schepartz.
Well, thank you very much. I'm delighted to be here. It's my first meeting, as Sangeetha described. And it's just wonderful to meet all of you. And I've learned so much about creatine deficiency diseases and so many things that I will have to think about, hopefully in the future. I'm looking forward to the next symposium and for continued interactions with all of you. Okay, so our lab works on a lot of things, but one of the things that we've been working on for at least a decade, if not more, is trying to achieve the elusive goal of delivering proteins directly into cells, into the cytosol, and into the nucleus, where they can act as replacement enzymes or alternatively as inhibitors of various therapeutically relevant processes. So the proposal that I submitted to ACD for the Race for the Cure Award was a strategy, a novel strategy that we delivered that we hope will enable the direct and dose-dependent delivery of GAMT to liver cells. But since the award was issued on the 1st of June of this year, I don't actually have that many results to share with you. But what I did want to do is provide you with an overview of what we've learned over the past 10 years about how to deliver proteins, active proteins, folded proteins to the cytosol and nucleus at levels that would allow them to actually act as replacement enzymes. And so I'm hoping you'll indulge me with some biochemistry and chemical biology and learn a little bit about why fundamental studies in cell biology and biochemistry are so enabling for the development of new therapeutic modalities. Okay. So first, some context. Okay, so we're trying to deliver GAMT, and one of the things you might be thinking about is I haven't heard any talks today in which someone's actually trying to deliver an intact protein for a therapeutic benefit. And I just wanted to put this work in context, because the truth is that there are lots of proteins that are well known. There are lots of proteins are used as drugs, and this has been true since insulin was approved for the treatment of diabetes, many decades ago. There are many examples of replacement enzymes that are used to treat multiple genetic disorders, some of them even in children from birth. The difference between all of those approaches and the approach that we are studying at UC Berkeley is that all of the examples to date in which intact proteins are used as drugs are examples where the function of that protein is required either in plasma or blood, or on the cell surface, or within little compartments in cells that are known as endosomes. And so the ways that proteins get into endosomes are very straightforward. The endocytic pathway, which includes endosomes, is one of the ways that the cell surveys what's present outside of the cell. And so the process of endocytosis involves literally taking little snapshots of what's present on the cell membrane and figuring out whether it's good or bad. And in fact, that is the fundamental process that leads to the immune response. Okay, so there are lots of proteins that are used as drugs that help millions of patients today, but they're all limited to these compartments that aren't the cell cytosol or the nucleus. And so what we discovered actually at Yale, where I was on the faculty for 30 years before I moved to UC Berkeley, is that at Yale, we discovered a way to help proteins find a way to escape from these little bubbles that are inside cells called endosomes to reach therapeutically relevant concentrations in the cytosol and nucleus. And so the origins of my being here really began with an email that I received more than a year ago from Sangeetha and Kaluna and Heidi and others that basically had read a paper that our lab had published in a really terrific chemistry journal known as ACS Central Science. And they had read this paper and they had realized that the implications of this paper were that the strategy for delivery that we had been working on could actually be useful for diseases that are related to creatine deficiencies that all of you care so much about. Okay, so the result of that first phone call was that I actually thought about all of the creatine deficiency diseases, and it turns out that GAMT has every single criteria that we know should make it easily deliverable by the delivery strategy that we have studied. And so I wrote a proposal, and the proposal was funded. It began on June 1st, and I have a fantastic graduate student at UC Berkeley, Jessica Murray, who's already begun to do experiments. But I can't tell you about the results because it's only been how many days? 27 days. So I don't have that much to share with you about the actual results, but I thought I would tell you a little bit about what this delivery strategy is, how it differs from really 35 plus years of other people trying to deliver proteins into cells effectively. And I hope you will recognize from the data that I'll share with you why GAMT is so perfect for this particular delivery strategy. Okay, so first some basic history. It's been known for over 35 years that in fact, if you take cells that are in culture and add certain proteins to the media, in certain cases, you can detect the activity of that protein inside the nucleus. And so that statement arises based on 2 papers that were published in the journal Cell in 1988, which was the year I began as an assistant professor at Yale. That's how many years ago it was. And just to make it clear, Cell is what I would refer to as an A+ journal in science. And so these 2 papers pretty much reported the same thing. What they described is that if they had cells in culture and they simply added to those cells in culture a protein known as TAT, which is made by the HIV virus, that what they observed in those cells is evidence that TAT had somehow figured out a way to get across the cell membrane, to get across the nuclear membrane, and activate genes inside the nucleus. And so that was the result of these papers that were published in Cell in 1988. Now, the workers didn't actually realize it at the time, but it took only about 3 years for one of those scientists, Alan Frankel, who's actually one of my colleagues at UCSF, was able to show, or was pointed out, that this finding, that TAT could somehow find a way to circumnavigate itself from outside the cell all the way into the nucleus, was actually some evidence that TAT could be used to bring other proteins, not just itself, into the cytosol and nucleus of mammalian cells. And hence the field of cell-penetrating peptides was born. Okay. The problem is that it basically has failed. So it's been 35 years since those 2 papers appeared in this A+ scientific journal Cell, and there have been hundreds of labs, tens of thousands of different variants of TAT that have been made and attached to all sorts of different cargo, but there have been no approvals. Okay, and in fact, if you go to the website for clinical trials, there is not one example of a molecule being delivered using a TAT-derived material. And so the question is, what can we learn from all of the research that has been done over the last 35 years to try to rescue this strategy? Because it is in fact true that TAT makes it to the nucleus, but we don't know how, we don't know why, we don't know how well. And the question is, if we can look back on those results, can we learn enough so that we could actually design a molecule that is as effective, if not more effective than TAT, at reaching the cytosol and the nucleus? Okay, so what has been learned? The first thing that's important to point out is that peptides are terrible drugs. Okay, they get cleaved in cellular media by lots of enzymes into little pieces, and as a result, it is very hard to correlate the amount of peptide that you're adding to the amount of activity that you see, since in many cases, you may be looking at the activity of a completely degraded material. Proteins, however, folded proteins, are much less sensitive to this degradation, suggesting that small proteins might actually be better suited as therapeutics than longer peptides. Okay, the second thing is that even over the course of 35 years, what most people measure when they add proteins to the media surrounding cells, or what most people measure when they inject a therapeutic into an animal, is they measure activity. Activity is not the same as delivery. Delivery is, I add this many moles, and I get this many moles in the cytosol. That is a delivery efficiency. If you measure activity, especially if your assay for measuring activity is amplified, so your signal is huge, even if the amount of material is tiny, you can really be fooled by an activity assay, which would allow you to believe that your molecule is present at a reasonable concentration when in fact its concentration is very, very low. And that is especially true when you're delivering molecules that are not catalysts. I think the third thing that we learned from thinking about this literature is that very few of the previous studies focused on understanding the how and why of how molecules circumnavigate within a cell without a transporter. Like, what are the factors that are required? What membranes do they have to get across? What are the criteria by which those membranes are crossed? So there was very little biochemistry and biophysics. In addition, there were really two assumptions in the field that in retrospect are really the most ludicrous. The first is, what we realized is that if someone made a delivery vehicle, whether it was a peptide or a protein, that seemed to work in one case, the assumption was that it would work in every case, regardless of the structure, the size, the physical properties of the material that delivery agent was trying to load into the cytosol nucleus. And of course, that makes no sense at all. And the last thing that we realized, which I have to admit we were ourselves guilty of, is that it turns out that many delivery strategies make use of the endocytic pathway that I described a moment ago. They make use of the fact that the cell surveys what's on the cell surface by taking portions of the cell membrane and turning them into little bubbles that are inside the cell. It turns out that those bubbles, one of their roles is to degrade those materials for purposes of the immune response. And if they degrade those the way they degrade those materials is by progressively lowering the pH of the liquid inside those vesicles so that it's very acidic. It turns out that many proteins won't survive if they're placed in an environment that is very acidic. And so the biggest problem, and the biggest problem we had to solve in the strategy that I'll describe to you, is that since we're using the endocytic pathway and we're using a new strategy to escape from endosomes, we have to make sure that the molecules that are attached to our delivery agent are not being degraded or denatured in this very acidic environment before they reach the cytosol. Because we can deliver as much as we want, but if the molecule that reaches the cytosol can't carry out its activity, like adding a methyl group to guanidinoacetate to make creatine, if the enzyme can't do that, there's absolutely no purpose in delivering the enzyme. Okay, so it turns out that, maybe 15 or so years ago, completely by accident, we discovered a small protein that appeared to be able to circumnavigate itself and itself attached to other molecules from the inside of endosomes into the cytosol nucleus. It's a very small protein. It's called ZF53. It's actually a single domain zinc finger for those of you who are familiar with molecular biology. So it's 28 amino acids long. And what is distinctive about it is that it has 5 arginines on the outside of an alpha helix. It turns out that those 5 arginines are essential for the first step in the process, which is gaining entry into the endocytic pathway. We do not actually believe that they are critical for the most important step, which is endocytic escape, which is escape from endosomes. So the second thing that we did after the discovery of this molecule is that we worked out a new strategy to actually understand and define exactly how much material was reaching the cytosol nucleus. It's a very fancy spectroscopic technique called fluorescence correlation spectroscopy. It's called a single molecule technique. And what it does is it actually tells you exactly the concentration of the molecule that you are looking at in the cytosol nucleus of a cell. So it's done with a microscope. And what's really fantastic about it is that it also tells you what its diffusion constant is, which gives you a sense of its molecular weight. And so why is that important? Because that's the first clue that what you're actually delivering is an intact protein and not a degradation product, because of course the degradation product has a different molecular weight, and so its diffusion constant is going to be different. So it's the first clue that we have that what we're actually delivering into the cytosol nucleus is an intact protein or intact enzyme and not a degradation product. So what we discovered using this tool is that most of the molecules that people call cell-penetrating peptides are not cell-penetrating at all, but our molecule is, and it can reach the cytosol nucleus with an efficiency of about 50%. It depends a little bit on what the cargo is, but the efficiency is very high for certain cargo, and we'll talk about that in a moment. And moreover, it can establish concentrations in the cytosol nucleus that are greater than 500 nanomolar, which is certainly the range that is needed for the delivery of a therapeutic enzyme replacement for therapeutic enzyme replacement. Okay, we've discovered that this molecule is not subject to degradation. It's actually incredibly stable. We've shown that it doesn't induce damage to endosomes, which would be a bad thing. We've discovered that the mechanism by which it escapes endosomes involves a natural process in cells that's called endosomal maturation, and that differentiates this method from many of the peptide-based delivery methods that have been studied before. And in fact, over the years, we and others have used it to deliver a small host of proteins, transcription factors, and nanobodies into the cytosol nucleus, including the transcriptional repressor / activator that is depleted in patients with Rett syndrome. Okay, so now I'm going to tell you what we've learned about how ZF53 operates. The first thing that we've learned is that because it has such a strong binding site for zinc, it is completely well-folded between pH 5 and pH 8. So pH 7.5 is the pH of the cytosol. In the endocytic pathway, the pH begins at about 6.5 and goes down to about 4.5. And ZF53, one of the reasons it works so well is because it remains folded in plasma, it remains folded in serum, it remains folded at the very beginning stages of the endocytic pathway. But then when it reaches a pH of about 4.5, it begins to unfold. Okay, and we can detect that either by CD or by NMR. It turns out that this unfolding event, the fact that ZF53 releases zinc and unfolds at pH 4.5, is absolutely essential for its ability to escape endosomes. And we believe that part of what happens is that when it unfolds, it associates with a lipid that happens to be enriched on the inside surface of those endosomes. And when endosomal maturation occurs, which involves a lot of scrambling of lipids between two different endosomes, that is the process by which ZF53, with or without cargo, escapes from inside endosomes into the cytosol, where it easily can reach the nucleus. We've also learned a lot about the cargoes that are suitable for ZF53. Not everything is suitable. I hate to say, I think it's very unlikely that the transporter for creatine is going to be suitable for delivery by ZF53. It's just too big. But GAMT is not. Okay, so what we've learned about this ZF53 mediated delivery is that it is incredibly well suited to proteins that are intrinsically disordered, such as the translation factor that's involved in the etiology of Rett syndrome. It's incredibly useful for proteins that are able to unfold at low pH so that they also can easily escape endosomes. And it's also good for small proteins, even if they're well folded and don't unfold in late endosomes. And so this data shows that we can deliver proteins as large as 35 kilodaltons or nanobodies that are super small. They are, in fact, even if they don't unfold, they're perfectly suitable for delivery by ZF53. Okay, so now let me get to the most important part of the talk, and that addresses the last criteria, or the last thing that we learned when I described all of the things we learned by thinking about why all of these previous delivery strategies had failed. And that is that the endocytic pathway is not a friendly place, right? As I said, as you go through the endocytic pathway, the pH gets lower and lower, and that can degrade proteins. It can also cause them to unfold, often irreversibly. And if a protein unfolds irreversibly, even if it makes it into the cytosol, it's not going to have the ability to carry out the catalytic activity that you so desperately wanted to be able to exhibit. Okay, and what I haven't told you is that when we take ZF53 to deliver the enzyme known as dihydrofolate reductase, which is not relevant to a creatine deficiency disease, it's actually not relevant to any disease, but it happens to be a very pH labile protein, ZF53 can move dihydrofolate reductase into the cytosol at levels of 500 nanomolar, like incredibly high levels. But that DHFR is dead as a doornail, right? Because DHFR, before it got to the cytosol, it unfolded in late endosomes irreversibly, and even though ZF53 was able to transport it into the cytosol, under those conditions, the DHFR could not refold in order to carry out the catalytic activities that DHFR is responsible for. Okay, so we got to work. And in fact, it wasn't me. I have to say this is all due to an incredibly talented graduate student, Angel Vasquez, who's now a postdoctoral fellow at UCSF. So Angel realized that although we had studied one family of zinc finger proteins, there's actually a ton known about zinc finger proteins because they were actually used as the predecessors to gene editing to generate, zinc finger binding nucleases that formed the basis for many companies. So there's a ton of literature about zinc fingers. And it turns out that the zinc finger that's present in ZF53, the way it binds zinc is to use two different types of amino acids. It binds two cysteines and two histidines. And in fact, it's the histidines that allow ZF53 to unfold at low pH, because at low pH, the histidines are protonated, the zinc dissociates. But there are other zinc fingers that don't use two histidines and two cysteines, right? Those would, ZF53 is a CCHH zinc finger. There are also zinc fingers that are known as CCCC zinc fingers, in which the two histidines are just cysteines. So they bind the zinc with 4 sulfur ligands. Okay, what this slide shows, okay, just to explain, is that it's giving you the affinity for, it's giving you the affinity for zinc as a function of pH. And so what you see is that the curve for the CCCC zinc fingers is shifted to the right. So what that means is that the CCCC zinc fingers should unfold at higher pH than the CCHH zinc fingers. So why is that good? Because if we have a delivery agent that has a CCCC zinc finger, it should unfold earlier along the endocytic pathway at higher pH, our hypothesis being that under those conditions, we would be able to deliver more acid labile proteins than we could deliver with a zinc finger that unfolded at lower pH. Because if a molecule is stable at pH 5.5, but not stable at pH 6.5, at pH 4.5, then this new zinc finger should be able to deliver them. And in fact, that is exactly what happened. So Angel designed a second generation delivery agent that we call AV53 for Angel Vasquez. And so the only difference between it and ZF53 is that it uses 4 cysteine ligands to surround the zinc, and it unfolds at pH 5.5, whereas ZF53 with 2 histidines and 2 cysteines unfolds at pH 4.5. So the question is, does it work? So the first thing we asked is, does it deliver? Okay, and that's absolutely true. What we find is that AV53 can make it into the cytosol just as well as ZF53. So changing the ligands surrounding the zinc doesn't affect in a substantial way how much is taken up into the endocytic pathway or how much of the material makes it out of the endocytic pathway into the cytosol. So that's what we could show. And then we began to look at whether or not it could actually deliver a functional acid-sensitive protein. And of course, we chose dihydrofolate reductase as a proof of principle because we already knew that ZF53 was able to deliver dihydrofolate reductase, but it was completely dead. Okay, so dihydrofolate reductase is a reaction, okay? And it turns out that it is an absolutely essential enzyme for cells to live. If your cells do not have if the cells do not express dihydrofolate reductase without the addition of supplements, they do not live. And in fact, that's what you can see here. So we're using cells here, Chinese hamster ovary cells, in which the gene for dihydrofolate reductase has been knocked out. And so what you see here, without any supplements, that's what the 0 means, over the course of 72 hours, those cells simply die. We can keep them alive by adding 2 of the materials that dihydrofolate reductase would generate in cells, just bypassing the need for dihydrofolate reductase. And when we add a full titer of those 2 supplements, you can see that the cells continue to live. That's why this line is flat. So if we take those cells And to them we add dihydrofolate reductase that is linked to ZF53, which we know is not able to deliver a functional protein, what we see is that those plots look exactly the same as they look like in the absence of the added enzyme. So this plot shows cells that have been treated in the absence of ZF53 dihydrofolate reductase. These are cells that have been treated in the presence of ZF53 dihydrofolate reductase. And you could see that they still die over the course of 72 hours. If you add a full course of supplements, right now they stay alive. Okay, no change, which is exactly what we would expect based on previous data. But now let's see what happens when we add AV53 linked to dihydrofolate reductase. So now what you see is that in the complete absence of supplements, now I can't tell you that the cells are perfect, okay, because there is a decrease in viability over the course of 72 hours, but it's substantially lower by a significant margin than what we see when we deliver dihydrofolate reductase with ZF53. And most importantly, in cases where we add supplements, now the cells are really happy, as indicated by the fact that they're beginning to proliferate. Okay, and so this data and other data that I haven't been able to show is consistent with a model in which AV53 allows delivery from earlier stages in the endocytic pathway, stages at which we can stop the acid-promoted denaturation of dihydrofolate reductase to the extent that we can compensate a genetic knockout in this cell line by simply delivering it linked to AV53 as a conjugate to AV53. So this is the paper that caught the eye of ACD. And this is the paper that led to my proposal to use AV53 to deliver GAMT to liver cells. Right, so the general idea is, let me explain why GAMT is so perfect. It is small, it is a single domain protein, it doesn't bind any metal ions, and we've actually already demonstrated that it is reasonably resistant to low pH. And so that is perfect for delivery. And so Jessica Murray is now working on developing a conjugate of AV53 linked to GAMT, including variants that have mutations and variants that have inactive versions of AV53 and all the controls that we can think of. And we're simply going to ask whether or not we can deliver a therapeutically relevant concentration and activity of GAMT into liver cells, where we would hope in the fullness of time it could be used in animals and eventually, if all goes well in people, to replace a cytosolic enzyme that is otherwise depleted in patients with that particular version of a creatine deficiency disease. Okay, so that's really all the science that I had to share with you. I want to thank my lab for being so fantastic. And I also wanted to point out a face that corresponds to the science that we'll be doing, and that's this lovely graduate student, Jessica Murray. She's now a third-year graduate student at UC Berkeley. She got her bachelor's degree at Boston University. She's a fantastic scientist, and she's very excited to try this project and to do her very best to make it successful. So thank you.
We're going to do questions in just a minute, but I have two quick announcements. One, for those parents whose children are not going to NAC, you can go and pick your kids up so that they can have lunch. I also want to request some of the last tables, if you could move up front so camp counselors can come and get started on their lunch. Now we will get to questions, but one quick comment that I wanted to make. I am not a protein engineer, so really who I want to call out and credit with the discovery of your paper and making the connection is Dr. Bill Snyder. He's one of the consultants that we brought on specifically to think about this and think about how this could be addressed. So thank you, Bill. We're here today because of you. Okay, now we'll get to questions.
Hi, Alanna. Thank you for that interesting talk. I'm wondering, instead of changing the amino acid sequence to go to the AC derivative of the shuttle that you made, have you thought about just swapping out the metals to because that could also give you an extra spectroscopic handle.
Yes, it turns out that we did try to swap out the metals. The metal ions that you would use are either cobalt or nickel. We see no big differences in the pH dependence, and we see, in some sense, I would say poorer behavior overall of the cobalt and nickel coordinated zinc fingers.
Thank you. And also, I'm wondering if you chop off the polyarch sequence, does that eliminate the uptake?
Yes, if you chop off the polyarch sequence, or if you change those arginines, even 1 or 2, to residues that are not arginine, the molecules are no longer taken up very well into the endocytic pathway.
In my lab, we've put polyarch sequences on small molecules and seen uptake and delivery and also efficacy. I'm wondering if your approach is applicable to small molecule delivery.
There's no reason why not.
Thank you, Professor Alanna. I'm a parent of a child who has GAMT, and my apologies in advance. Everything you showed, I was not able to get more than 5%. It was a lot of chemistry for me. I was focusing as much as I could, but that's not the relevant point.
Ask anything you want.
Yeah, that's exactly where I'm going. So help me understand, as a parent of a GAMT child, how you are, so I understood gene therapy to an extent. How does it going to change the genes, and at a cellular level, it's going to create the creatine and reduce the GA level. How does your research and the therapeutic answer, is it going to be administered? Is it going to be once? Is it going to be multiple times a month? How does it change our current protocols based on this research?
That's a fantastic question, so thank you for asking it. I actually thought about trying to include that, but it just slipped my mind during the talk. So the big difference and the big advantage of naked protein delivery is that it's, in many ways, it has many of the same advantages of a small molecule or an injectable drug. So it can be administered in multiple doses, right? Because not all children are the same. Not all children will need exactly the same amount of GAMT to improve their symptoms. And so the advantage is that it can be provided in multiple doses. It also could be administered multiple times. We are delivering, much of what we're delivering is a natural enzyme. We will use the human version of GAMT, which will overcome many, perhaps not every, but many criteria that would result in immunogenicity of this delivered protein. So I think the advantage is that it could be dosed. The advantage is that molecules like this with lots of arginines, they all go to the liver. Okay, so, and we've actually verified that in animal studies, that these molecules all go to the liver. And so by delivering an active enzyme to the right cells in the liver, it would provide the enzyme that will lower the concentration of guanidinoacetate, which is good, and will raise the concentrations of creatine, which is good. And then the natural process by which creatine is secreted out of the liver finds its way to the brain, and the natural transporters will be able to allow it to transcytose and then get into all of the neural cells that it needs to get into. So I think that it is simpler, okay? It is likely to be less costly, but it's different. I think when you're dealing with diseases that are urgent, I think the most reasonable approach is to have multiple shots on goal. This is an alternative shot on goal to gene therapy. And at this point, no one can predict which one is going to be better and whether it's going to be better for all patients. And so I think having multiple approaches in general makes a lot of sense.
Definitely not my area because I don't have a child with GAMT, but just curious, is this, just so I understand, is this something that would be like a daily or weekly dose, or something that once it's there, it corrects permanently? I'm just trying to understand.
It will not be permanent. So I would imagine it would have a lifetime, much like other replacement enzyme therapies. It really depends. So I don't know what the answer is. It could be something that is dosed daily. It could be something that is dosed weekly. I think if we are very, very lucky, it would be longer than that. It really just depends, and there's really no way. I wish I could predict, but I just can't.
Thank you so much, Dr. Schepartz. This was excellent. This talk is recorded by Dr. Sara Biagiotti. She couldn't be here in person today. She is the assistant professor of biochemistry at the University of Urbino, where she leads research on red blood cell-based platforms for therapeutic delivery. She began working on RBCs as drug delivery systems over a decade ago, and she is now the principal investigator of a next-generation EU-funded project developing mRNA EV therapeutics platform specifically for GAMT deficiency. She is also an ACD fellow from this year's cohort. Good to go.
Or good afternoon, everyone. I don't know what time is now in the United States. First of all, I would like to thank the organizers for the opportunity to share with you our project, my project, and my data. And secondly, I want to apologize for not being able to be with you in presence today. But I'm not exactly behind the corner. I actually live in Europe, in particular in Italy. I live in a very small town in the middle of Italy, and I work at the University of Urbino. As you can see, Urbino is a very beautiful place that is our heritage site. My group at the University of Urbino has been working on red blood cells, these ones, as drug delivery system for more than 30 years. These are some of the molecules I have loaded and delivered during my life, and they comprise either small molecules or biologics such as recombinant proteins, short and long RNAs. Here you can find some references. Okay, currently we are using RBCs to set up different strategies for several rare diseases. For example, in ataxia-telangiectasia, we are using RBCs to deliver for the slow delivery of dexamethasone. And this was a very lucky case because we reached the phase 3, so the last stage of the clinical research. Here you can find a very nice reference. For what concerns phenylketonuria and GAMT deficiency, we are using RBCs to load enzyme. For example, in the case of PKU, we are loading the PAL, while in the case of GAMT deficiency, we are loading the GAMT enzyme. In those cases, we are at the preclinical stages. To go more in detail for the GAMT, in 2022, we proposed the first strategy, which is an enzyme replacement therapy. So we load our RBC with a recombinant fort of GAMT enzyme. Our RBC with the enzyme inside goes in the circulation, acting as circulating bioreactors, where the guanidinoacetate enters the cell, is being converted into the creatine that can go out and reach the circulation. However, we found that this system was not so effective. Why? Because this metabolite, this adenosyl methionine, was limiting in RBCs. So we had the idea to load a second enzyme, which is MET, methionine adenosyl transferase, to increase the amount of SAM. The system worked very well in vitro, but when we went to mice, we had some troubles. So we changed our strategy. A few years ago, we moved from a cell-based to a cell-free drug delivery system. In particular, we moved to RBC-derived extracellular vesicles. Extracellular vesicles are very tiny particles, like these ones, that are surrounded by a membrane bilayer and that can be loaded with several drugs inside. And the big advantage is that they can reach the recipient cell and discard the cargo directly inside the recipient cell. RBCEVs are very promising for several reasons. because they are safe, completely safe, non-toxic, biocompatible, non-immunogenic, and so on. They are very easy to obtain on a large scale and at low cost, starting from blood. Blood that can be collected from patients or donors. And finally, they are highly versatile because they can load it with a lot of drugs, as I told you. They can be used fresh to store, blah, blah, blah. But most importantly, they can reach all the body compartments. In 2023, we developed and patented a new method to produce RBCEVs already loaded with the RNA cargos. This procedure is a three-step procedure. The first step is the loading of the cargo RNAs into the RBC. Then the second step is the vesiculation by a physical method we patented to obtain very good RBCEVs with a high amount of RNA inside. The third step is the purification and the characterization of the final products to confirm that we were actually able to obtain our extracellular vesicles with the membrane, with the surface antigens, and absolutely with the cargo inside. The patent has been already accepted in Italy and is on the way for the international application. Sorry. Okay, this is the first publication, the proof of concept that our system could work. We used our RBCEVs to deliver a short RNA, a microRNA, sorry. The microRNA in question was the MIR210, and in this way we were able to demonstrate that our procedure is reproducible, the loading was efficient, as well as the release of the cargo inside our cells, and once reached the recipient cell, the cargo was effective, so it was able to exert its biological effect. Here you can find the reference. Okay, then we moved at preclinical level with this project. The title of the project is the RNA, and is to set up an RNA-based therapy to treat GAMT deficiency. The therapy is obviously based on RBCEVs. This project has been supported by the European Union, and this project has been completed now. First of all, our murine model is the knockout mouse that has been set up in 2004 in the medical center in Hamburg. We have this model available since 2018, and we also characterize very well this murine model, and we can say that it can recapitulates very well the biochemical phenotype, while regarding the behavioral phenotype, we have only mild to moderate cognitive or motor impairments. Okay, this is the graphical abstract of our project. Once again, we have 3 steps. The first step is the production of our vesicles, our RBCEVs, loaded with a synthetic form of messenger RNA encoding for the GAMT enzyme. The second step is the administration of our vesicles in the murine model in knockout mice. And finally, the third step is the evaluation of the efficacy at both the biochemical and the behavioral feature levels. Okay, so the first step was the mRNA synthesis. Together with GScript, we designed in silico our synthetic messenger RNA. First of all, we used all the tricks used to increase the RNA stability, such as pseudouridine modification, but also to improve the protein expression, CAP1, poly-A optimization, blah, 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 as has been already used for vaccines, Pfizer, Moderna, and so on. While regarding the coding sequence, we use the coding sequence of our GAMT protein. And finally, we decided to optimize the codon to allow the translation of our mRNA, both in human and mouse hosts. A brief focus as CDS, as coded sequence, we use the one of the recombinant protein we used in the paper in 2022, as I already told you. This protein was engineered to increase its stability and enzyme activity. So it was more active than the native one, our mutant protein. Here you can find the 4 mutagenic sites we put in the sequence. Okay, the second step was the setting up and the optimization of the RBCEV production. RBCEVs loaded with our in vitro transcribed GAMT mRNA, so our synthetic messenger RNA. First of all, we were able to optimize the loading of this synthetic mRNA in the RBC, in the cell. Most importantly, the RNA cargo was efficiently retained in the final vesicles. We used absolute quantitative PCR to evaluate, to measure the amount of RNA, and as you can see, it was very, very high in both the steps, with the final step having 2 micrograms of RNA per milliliter of preparation. I took the opportunity to say that from the first paper, now we, in this case, we had a lab scale production, while in this case, we moved to we scaled up the procedure to go in at the preclinical level. Okay, regarding the vesicles, we characterized physically those particles by transmission electron microscopy to demonstrate that they are rounded, lipid bound, and so on. And we characterized the size of the population. So as you can see, we obtain a homogeneous population of vesicles of RBCEVs with a size mode of 150 nanometers and a very good yield. So we were ready to go into mice. Okay. Oh, not yet. Before going into mice, we wanted to confirm that our strategy could work at least in vitro. So we selected a kind of cell that has a little small amount of endogenous GAMT mRNA, as you can see. We treated those cells with our vesicles containing the synthetic one, the in vitro transcribed GAMT mRNA, and the treatment went on for 1, 2, or 3 days. And as you can see, the cargo RNA was actually released in our cells after 1 day. Regarding the protein, it was actually translated into the respective protein. In this case, we had the maximum overexpression after 3 days. Okay, now we are actually ready to go in mice. Okay, first of all, good morning or good afternoon, the organs. In this way, we demonstrated that they have a natural liver tropism, so they were mainly recruited by the liver, and we were very happy of this because, as you know, the GAMT enzyme is mainly active in the liver, while the spleen has been performed in collaboration with the National Institute of Health in Rome with Dr. Sella Canese, and what we saw is that by proton MRS, we are talking of the brain, okay? By proton MRS, we had an increase in total creatine, so creatine plus phosphocreatine, while by the phosphorus signal, we found a an increase in phosphocreatine and a corresponding reduction in phosphoguanidinoacetate. Okay, this is a focus on the phosphorus signal because it's more pure, because as you know, the phosphate groups, the metabolite with phosphate groups in the cells are not so much. We have the 3 peaks of the ATP, the 3 phosphate groups of our ATP, and here you can find the peaks of phosphoguanidinoacetate or phosphocreatine, okay? Let me pay the attention on this peak. And on the quantifications, sorry. And as you can see, both in the brain and in the muscle, we were able to reduce the amount of phosphoguanidinoacetate, okay, and contemporarily to increase the phosphocreatine, okay. This effect was more prominent in the muscle, but this is quite normal because we thought that is needed more time to change the equilibria in the brain, the equilibria between phosphoguanidinoacetate and phosphocreatine. Okay, in parallel, together with our colleagues at the Sapienza University in Rome, they are the psychobiologists, we are profiling the behavior of our mice. We performed a lot of tests, both during the development and in the adulthood. But as I told you at the beginning of my talk, the impairment at the biochemical, the behavioral level is quite mild. Actually, as you can see, all the reflexes are delayed, however, the milestone, the developmental milestone are regained during the development, the day life, okay? Also for these tests. The only test that was very, very different was the ultrasound vocalization. So it was completely different between wild type and the knockout mouse, but usually this vocalization is lost during adulthood, so is not so useful for us. The only test we found significantly different in the adulthood is the social interaction. So our knockout mice do not interact with other animals, okay? So in the future, we could use this test for to assess the functional rescue after our treatment. Okay, in the last part of my talk, I would like to present the current project that has been funded by the Association for Creatine Deficiency under the Fellowship Program 2026. Okay, this is the title of the new project. Okay? And it was very important for us because it allowed to go on and hopefully finish our preclinical assessment. So the preclinical assessment of our strategy. Okay, this is the study protocol design. So we increased the number of animals treated and also the controls. We maintained the dose, 200 nanogram dose, and the interval between 2 different treatments, that is 7 days. But we are trying to optimize all the steps in the middle, starting from the result we observed in the pilot study. Okay, I took the opportunity to say that these analysis, the dried blood spot analysis, are performed together with the National Newborn Screening Center in Rome. Okay, at the end of the treatment, mice will be sacrificed, the organs will be collected. So during the treatment, we will perform dried blood spots, but at the end of the treatment, we will measure the metabolites also in the brain and in the muscle. Okay, so we are in the middle, okay? So just to show you, that we are trying to, we are optimizing the procedure, trying to reduce the intervariability between a treatment, between the spots, and so on. We are on a good track. Also, we decide to perform MRS analysis. Also, even if there wasn't inserted in the project at the beginning, because we saw that they are very, very important. So this is the experimental timeline. We started our treatment a little bit on late. So we started at the beginning of May. We will finish our treatment at the end of July, and then we will do all the analysis, and I hope to have the data by the end of September. Okay. Last but not least, I would like to show you what's new. So we are currently trying to compare the efficacy of two different RNA molecules. When we started the project, the first one project, the linear was very new, the linear mRNA. So as I told you, the one that has been used for vaccines in the pandemic. During these 2 years, the circular RNA went out, okay, came out. And they are very promising for several reasons. First of all, because they have a longer life with a stronger protein expression, and so we can reach the same effectiveness with a lower dosage. In the right, I am showing you a pilot study using 2 different RNA molecules, one linear and one circular, coding for a green fluorescent protein that is very, very used in the lab. And we are comparing the protein amount after 6 days. And as you can see, starting from the same amount, at the end of the 6th day, the circular one is at least 4 times more expressed than the first one. So it is very, very promising. Okay, I finished. These are the people, some of the people I want to thank. This picture has been taken a few days ago when we met at Urbino at our campus. With some colleagues of the University of Rome, just to discuss the data I showed you today. Obviously, I would like to thank the association for this big opportunity, and finally, you for your attention.
We're gonna get ready to start off our afternoon sessions. To start off, I'd like to welcome Melissa Evans. Melissa is part of our Parents 2.0 cohort. She's an award-winning healthcare executive and most importantly, mom to Logan, who was diagnosed with CTD following a cardiac arrest and full genome panel at age 10. Melissa and Logan are advocates for creatine and brain health awareness. They've been featured in the Washington Post, local news, as well as magazines. She served as a keynote speaker at the Children's Ball in Washington, D.C. in 2026, earlier this year, and helped raise $2.6 million for the hospital that saved her son's life. Melissa, thank you so much for using your voice to raise awareness about CTD and CCDS in general. We're so happy to have you share your story here with us. Over to you.
Thank you. So again, I'm Melissa Evans, and I'm a healthcare executive, CEO. But most importantly, as some of you may know, I'm an amazing mother to Logan, some of the time. I love the fact that Logan has brought me a greater awareness of rare diseases, especially creatine deficiency. And I'm excited to share the video that was created for my keynote address at the Children's National Gala in April. I'm going to make some remarks after the video is shown. So thank you. Okay, no worries. Different schools. All right, thank you.
We adopted Logan when he was 3.5.
We saw so much potential in him.
But it was quite difficult.
He had no social skills. He was highly aggressive, was kicked out of 8 different schools. He was listed on his IEP as emotionally disabled. December 2021, our lives changed. Jason called me and let me know that Logan was pretty sick. He had emesis, which is uncontrolled vomiting, for 2 days. We took him to the local emergency room. They didn't know what was wrong with him. They transferred us. As soon as we got to the big hospital, they got Logan on a helicopter and brought him to a liver transplant unit. They tried many different things. They had to put him on a ventilator because the rest of his body was shutting down. Christmas night, they called us at 2 a.m.
We thought he was dying.
Why was a child having a cardiac arrest for no apparent reason?
And then they said that they can't do anything with the heart. I mean, it just was the worst day of my life.
Miraculously, the medical director called, and Children's National brought their entire team to Georgetown and stabilized him on Christmas night and was able to save his life.
Yeah.
I met Logan when he was 9 years old. He was in our cardiac intensive care unit. It was really interesting because he did not have any past medical history. So my first thought was that there's definitely something else going on.
When we got to meet him, he came to our research program to say, could we find out what was going on? We were able to look at his entire genome.
A couple weeks after I get home, I get a phone call from Dr. Berger's office. And they said, we have figured out what's wrong with Logan. He has a very rare disease. It's called creatine deficiency transmitter disorder.
In terms of Logan's care, getting this diagnosis for him was crucial. In his case, we were able to actually do RNA sequencing next on him. Logan's testing actually suggested that there was actually hope for treatment.
We didn't notice the changes right away. The school, they said, who is this kid? Like, this is not the same Logan. And he started learning for the first time at age 10.
My name is Logan and I am 14. My favorite things to do are wine pie. I love Boy Scouts.
Logan's life has completely changed since he's been on the treatment for creatine deficiency.
I love seeing Logan in clinic and hearing from his family. He is thriving and he's doing great in school. Patient care is such a team sport. We really rely on families and physicians across all of the divisions, and we rely on our donors and the support of the community.
Genetics is a really evolving field in terms of the impact we actually have. There's such a short distance between getting to that diagnosis, figuring out the right treatment, and there's so many families that just can't get there without the support of our donors here at Children's National.
Children's National saved my son. Children are the future. And if we can help solve the mysteries of some of these illnesses that are out there, the world would be a better place. Thank you. When I watched the video, I cried the first few times I watched it, but then I realized these were like tears of joy. So on behalf of my family, I want to express our extreme gratitude to the ACD, the researchers that are here, and the families that are supporting their loved ones with creatine deficiency. As mentioned earlier, the gala raised over $3.6 million for the hospital's rare disease program. We are happy to support a few people in the room from Children's National. So as you saw, we came this close to absolutely losing Logan. And in the depths of our nightmare, we truly did not know what to expect. But research literally saved his life, and the care that he got gave him a future. His chance of finding like the right treatment, the right supplements, was probably like less than 1 million. But when we arrived at Children's National, the right people came together at the right time. And Logan has had several like articles written about him, ranging, like from being case 3.3. But, I know him as my amazing son. But my favorite article that was written about him was the Mother's Day issue of the Washington Post, was actually highlighted how whole genome sequencing has saved families. And I think that's just amazing, because that's what all of us families want to hear. We truly received the gift of a lifetime when he got the CTD diagnosis. And that gift probably would not have been possible without some of you in the room, so thank you. But I'm also glad to know that his donated cells are being used to move science forward. Logan got a kick out of that, finding out his cells are out there and being used to help other people. But as a closure, Logan actually wrote some remarks for me to read. He's pretty shy around large groups of people, but not around small groups. So if you want to meet him, he'll be, of course, around again tonight. So what they did also at the Children's National Gala is they created these bookmarks to give out to all the guests. So I'm just going to read his remarks that he wanted me to share. It says, meet our Children's Ball patient ambassador, Logan, age 14. Hi, I'm Logan, and I'm passionate about Boy Scouts and love to ride my bike. For the first 9 years of my life, I struggled with behavioral problems and learning in school. Then on Christmas Day, I almost died from a cardiac arrest. Everything changed when I was transferred to Children's National. Doctors found out I have a rare metabolic disorder called creatine transport deficiency, one of only 300 cases in medical history. I got treatment at Children's National, and now I thrive in school and in life. And that's his little picture and card. So thank you.